All right.
It's not working.
Cool.
The ones you guys made.
All right, I think we should begin. Everyone, hello, welcome. Welcome to your Physiology midterm review session. Okay, we are your Sci Peer Tutors. I'm Jose Vasquez, a neurobiology major, fourth year, minor in psych, and doing research at the School of Medicine, all kinds of stuff. You can read it on the website. And if you have any questions whatsoever about this or different subjects, especially in the biosci, feel free to reach out.
Hi everyone, my name is Monica. I'm also a fourth year neurobio major. What else? Oh, I'm an art history minor. Ariella, introduce yourself.
Hi everyone, I'm Ariella. I'm a fourth year as well, double majoring in public health and psych. And I just want to say thank you all for coming. We hope this is beneficial. Y'all are gonna learn a lot.
All right.
Before we begin, these are our office hours. If you're not already aware, how you can find us via Zoom links and our email information. And then this is actually something that we are requesting for you guys to, this is where you get to say what you have to say about us. Give us some good feedback, bad feedback. How did we do? Let us know. So once you're done with this review, if you don't mind taking the time to just, leave us an evaluation, it would be very beneficial so we can improve on the future sessions, okay? All right, I'll let Ariella take it from here.
All right, so the first topic we'll be covering over today is on the next slide, diffusion, membranes, and transport. All right, so first off, we're just gonna start off with homeostasis. So homeostasis is the body's self-regulating process in which biological systems tend to maintain stability while adjusting to both internal and external changes in conditions that are optimal for survival. So all living organisms, from plants to puppies to people, must regulate their internal environment to process energy and ultimately survive. All right, so fluid compartments. So there are two general fluid compartments. There's the intracellular fluid, or ICF, and this is the fluid within the cells. So it contains about 2/3 of the body's water and is generally high in potassium and low in sodium. And the other compartment is the extracellular fluid, the ECF. So this contains the interstitial fluid and plasma. So interstitial fluid is between the cells and plasma is just a fancy word. So the ECF contains about 1/3 of the body's water and is generally high in sodium and low in potassium. So in the picture, you can see that plasma is red, the purple is interstitial fluid, and the yellow is ICF. All right, moving to the next slide, osmolarity. So osmolarity is the number of solutes present in a unit volume of fluid. So I just found this really cute photo that gives you a sneak peek into osmosis, a type of passive transport. All right, Jose, if you could click one more time for me. And here's a table that compares hyperosmotic versus hypoosmotic. So if you could click one more time. So hyperosmotic is a higher solute concentration with low free water concentration. And if you could click one more time for me. And hypoosmotic is a lower solute concentration with a higher free water concentration. All right, so now let's go on to tonicity. So this is a physiological feature of a fluid that determines whether fluid will move into or out of a cell. So it's pretty much just the direction of osmotic water movement. So I found another cute photo that you can use to help guide you when you fill out this table on your own. Jose, if you could help me click it 2 times. So hypertonic solution. So this has a tonicity greater than that of the ICF. So osmotic pressure is unequal inside and outside the cells. So this type of fluid draws water out of the cells and into the more highly concentrated ECF. So this results in the cell shrinking. While hypotonic solutions have a tonicity less than that of the ICF. So osmotic pressure draws water into the cell from the ECF. So this causes the cell to inflate and they can eventually burst. And lastly, we have isotonic solution. And this is when they have a tonicity equal to that of the ICF. So the osmotic pressure is therefore the same inside and outside the cells. So they neither shrink or swell with fluid movement. So all these terms are just relative. That means they just describe how one solution compares to another solution in terms of osmolarity. For instance, if the fluid inside a cell has a higher osmolarity than the surrounding fluid, then the cell interior is hypertonic to the surrounding fluid, and the surrounding fluid is hypotonic to the cell interior. And to add on before we move on, I feel like it's very easy to mix up hypertonic with hypotonic solution. So a nice way to help me remember is when I look at hypotonic, I see the O, and I just think of how the cell swells into a big O. All right, so next up, I have a clicker question for you guys. So let's say Dr. Azizi places red blood cells into pure water. What is the blood cell and what is pure water? And what direction will the water move and what will happen to the cells as a result? And you can go ahead and type your answers into the chat, or someone can shout out the answer if they like as well. Any guesses? All right, I'm just gonna give a bunch of you guys time to think about it before we move on and give you the answer.
And anybody brave enough to speak their mind and explain their thought process or why they chose the answer they chose?
So the blood cells are hypertonic because pure water wouldn't have any like solutes in it, which is why the pure water would be hypotonic because it's less, the osmolarity would be less. And so water would move into the cell and the cells would burst because the water would move to the concentration with the higher solutes.
Yes, that's correct. Beautiful. She honestly couldn't have explained it any better. So the blood cells are hypertonic. And
my review session.
Oh, good. You can't hear it on the thing. You have to hear it on
All right, so moving on to calculating osmolarity. So this is the formula for calculating osmolarity. So its solute concentration is equal to the number of solutes divided by volume. And the units is in osmolarity over liters. So here's just a small quick question. What is 2 moles of NaCl equal to in terms of osmolarity? And what is 2 moles of glucose equal to in terms of osmolarity? Yes, Priscilla, that's correct. So moving on to the next slide. So how she probably calculated, I assume, is that NaCl fully dissociates in water, which leaves 1 mole of NaCl is equal to 2 osmolarities, and then just times it by 2. whereas glucose does not dissociate in water. So it just is, oh, it says 4, but it should say 2. Sorry about that. So NaCl is 4 and glucose is 2. All right, moving on to I have another clicker question for you. So what is the total osmolarity of a solution that is 2 moles of magnesium chloride and 3 moles of glucose? This one has a little bit of math in it. So I think it's more testing of your math skills.
All right, I'm seeing a lot of D. Does somebody want to share why they got D? All right, which is it? Denden, you want to share why you got C? Or why you
Okay.
think?
Yeah, I'll share. I'm not too confident in this, but okay, we know
That's
that.
fine.
Yeah, we know that osmoles are like our number of solids over a liter solution, right? So over here we have magnesium chloride. Say it disassociates in water.
Mhm.
I think we'd have
I think he's attending it.
1 magnesium and 1 chloride ion and another chloride ion. And since there's 2 molar of that, you just multiply those by 2. So that'd be 2 magnesium and 4 chloride. So far we have 6 solids in total. Glucose doesn't dissociate, so we'd add the 6 plus the 3. And that would give us 9.
That's correct. Good job.
Beautiful.
Yeah.
That is correct.
All right.
So just to reiterate, magnesium chloride dissociates in water, but glucose does not. All
Wow.
right, now let's move on to the next slide. So Fick's law of diffusion. So diffusion is just the net movement of a substance traveling down its gradient. So a concentration gradient is just a region of space over which the concentration of a substance changes, where it naturally moves from an area of higher to lower concentration. So Fick's law of diffusion states that the movement of particles, i.e. diffusion flux.
I'm so sorry to interrupt, Ariella. They're asking, there's a bit of confusion around the math of how you solve the last problem.
Oh yeah, of course, sorry. So for this question, we know that magnesium chloride dissociates in water into separate ions. So 1 mole of magnesium chloride separates into 3 in total, with the magnesium, the chloride, and another chloride. So we multiply that by 2, which is 3 times 2 equals 6, and then glucose does not dissociate at all, so we're just adding 6 plus 3, which is equal to 9. Does that help clear any questions? All right, beautiful. Yes, thank you, Jose. All right, and to continue, so Fick's law of diffusion states that the movement of particles or diffusion flux from high to low concentration is proportional to the particle's concentration gradient, the surface area of the tissue, as well as the solubility of the gas. And it is also inversely proportional to the thickness of the tissue. All right, so let's move on to membrane proteins. So, sorry, I just think these pictures are really cute. I don't know if you guys can relate, but Jose, if you can help me click 2 more times. So there are 2 general membrane proteins, channel and carrier. So to start off, channel proteins have the ability to form hydrophilic pores in the cell's membrane. It can transport molecules down the concentration gradient, and they can be open or gated. And if they are gated channels, they can open and close in response to either chemical, mechanical, or electrical signals. So examples of channels include ion channels like sodium or potassium, and aquaporins. While, yes, beautiful. Carrier proteins can transport substances across the membrane, both down and against the concentration gradient. So they can be uniport, which means they transfer only one substance. Symport, which means they can transfer two or more substances in the same direction, or antiport, which is when they can move two or more substances in the opposite direction. So examples of carriers include sodium-potassium pumps, glucose-sodium co-transport, and channel and carrier proteins both transport material at different rates. So in general, channel proteins transport much more quickly than do carrier proteins because they're just like simple tunnels. Unlike carrier proteins, they don't need to change shape and reset each time they move a molecule. Okay, and moving on, we have different forms of membrane transport. So the simplest form of transport across a membrane are passive. So passive transport does not require the cell to expend any energy and involves a substance diffusing down its concentration gradient across a membrane. So diffusion results from molecular motion and doesn't require ATP pretty much. So for example, in simple diffusion, typically small uncharged lipophilic molecules can diffuse across the lipid bilayer. And all other kinds of transport across the membrane involves membrane-bound proteins. For instance, facilitated diffusion requires a membrane protein. And osmosis is just a specific form of facilitated diffusion solely for water, and it works through aquaporins. And in active transport, unlike passive transport, the cell expends energy, for example, in the form of ATP, to move molecules or ions against its concentration gradient. So I have a clicker question for you all. Which of the following membrane transport methods do not require membrane proteins? A, active transport, B, facilitated diffusion, C, osmosis, D, simple diffusion, or E, none of them. Yeah, it looks like we're all on the same page. Beautiful. So simple diffusion is the only one that does not utilize a channel or carrier. The key word is simple. So I just gave you just a basic question as a warm-up. All right, now let's go on to feedback mechanisms and regulation. So the image on the left is just a picture of the general endocrine pathway, and it shows the general steps that occur. For example, if you touch a hot plate, this stimulus will be sensed by your body and lead to a response such as like flinching away. In addition,
sorry, Ariella. Oh, sorry. One more thing. There's a little bit of confusion about osmosis essentially requiring channel proteins. Someone is saying aquaporins. Sorry, a transporter. Using a transporter.
So osmosis uses aquaporin, and that is an example of a channel. Would anyone have any other questions? Okay, beautiful. Sorry, I should look at the chat more. Okay, so the pathway, it's just like the release of hormones can be effectively controlled by feedback loop mechanisms, which lead to the next slide, which are feedback loops. So feedback loops allow living organisms to maintain homeostasis. So positive feedback loop amplifies, while negative feedback loop inhibits the system. Okay, so now local communication. So this includes direct and nearby cell communication. So for direct communication, this includes gap junctions and contact-dependent signals. So gap junctions is when the cytoplasm is connected and there's a direct exchange of signal between these cells. Whereas contact-dependent uses signal molecules and receptors to directly interact and communicate with one another. And we also have autocrine and paracrine signaling, and this is nearby communication. So autocrine signal secretes information into the ECF and acts on itself, whereas paracrine signal secretes information into the ECF and acts on neighboring cells. So both of these type of signals depend on diffusion as a means to reach their target cell and differ primarily in their targets. So moving on to long distance communication. So how it works is it can occur through the nervous system or the endocrine system. So the nervous system communicates through neurons, which is the electrical signal, and neurotransmitters, which is the chemical signal. So this form of communication is fast and specific, while the endocrine system communicates through hormones, which are chemical signals, and this form of communication is much slower, less specific, and results in a long-lasting change. So I have another question for y'all. So hormones and neurohormones travel through what? And neurohormones and neurotransmitters are released by what?
Don't be shy. You can speak up if you'd like. You can unmute yourself. Some people in the chat. All right. What do you think, Ariana? Are they getting it?
Yeah, they are.
Good job. All right, everyone.
Yeah, you guys got it. So hormones and neurohormones travel through the blood, and neurohormones and neurotransmitters are released by neurons.
I see somebody put vesicles there. Dean, you wouldn't be wrong for saying vesicles, but there's a little bit more to it. I think that's a more precise answer, and it depends on what exactly you're talking about, what mechanism.
All right, so let's move on to hormones. So hormones are produced by glands, specialized cells, and neurons. So what they can do is pretty cool. They can alter the membrane's permeability and change the rates of enzymatic reactions. So they have like 3 different types of hormone interactions, which include permissiveness, synergism, and antagonism. So permissiveness is when one hormone promotes cellular response to another hormone. Synergism is when the combined effects of multiple hormones surpass the sum of their individual effects. And antagonism is when one hormone decreases the cellular response to another hormone. And tropic hormones is hormones for regulation in the endocrine system for hormonal release. All right, so there are 2 types of hormones, peptides and steroids. So this table just simplifies the differences. Jose, if you could uncover what's beneath those. Beautiful. So steroid hormones are not soluble in water, but soluble in lipids. They can penetrate the cell surface membrane and they form the hormones receptor complex in the cytoplasm. So steroids can activate genes and synthesize enzymes, which bring about changes in the cell. So next we have peptide hormones, which are water soluble but not lipid soluble. And it forms the hormone receptor complex at the cell surface membrane and activates a membrane enzyme, messengers, genes, and enzymes, which therefore bring about changes in the cell. Okay, so next up.
There's a question. Sorry, there's a question in the chat from Andrew. He's asking, do steroids enter the nucleus region / bind at the nuclear membrane?
Okay. So steroids, they can enter. So I think that goes on with this slide, which is the synthesis of hormones, which explains how they move throughout. So how about I go through the slide and see if this answers the question. All right, so for the synthesis of peptides, how it works is that it first starts at the ribosome where mRNA is turned into the pre-hormone, and then it transfers and moves to the ER where it is cleaved and changed into the prohormone. And then it moves to the Golgi's, and then it is cleaved once again into active peptide hormones and peptide fragments. And then in the ECF, it can be released and leads into the plasma where hormone circulates to its target. Whereas for the steroid, when it's made, it's initiated by hormones and it's formed in endocrine glands. And it's derived from cholesterol, and the hormone diffuses into the bloodstream. Awesome.
I just have one more thing to add on the steroid argument. So if you think about it, they would have to cross the nuclear membrane, because if you remember the comparison between peptide and steroid hormones, so steroid hormones are the ones that are going to enact change at the genetic level. Like if you guys remember, Dr. Azizi was talking about how they can enter the nucleus and make permanent alterations, right? So they're slower, but the effect is more prominent versus peptide hormones that are more quick, but the effect is not going to be as long lasting.
All right, thank you, Monica. So I have another question for y'all. So why do steroid hormones usually take so much longer to act on peptide hormones? So A, because steroids modify existing proteins. B, because steroids usually initiate new protein synthesis. C, because peptides usually initiate new protein synthesis. Or D, none, E, all. Yay, beautiful. The answer is B. So steroids usually initiate new protein synthesis, which is why it takes a bit longer than peptides to, which just modify existing proteins. All right, moving on to pathology. So these are just pretty much some vocab I'm throwing at you for this slide. So you should know the difference between hypersecretion versus hyposecretion. So hypersecretion is more excess hormone secretion due to 1, loss of feedback regulation and or excess hormone production. Whereas hyposecretion is less, so it's the loss or decline in hormone secretion due to deficiency of hormone production. So for target cell pathologies, there can be downregulation of target cell receptor and issues with the transduction of signal in target cells, which can lead to endocrine pathologies. All right. And now I'm going to hand off nerves to Jose.
Oh, hello everyone. Just a little nervous, but I think we'll get through it. This is my first time hosting a review session, so cut me some slack. Just kidding. You can be as mean as you need to be. Well, I'm going to begin with nerves. So this is just an overview. So when we share the slides, you can reference it. I'm not expecting you to read all of this right here, right now, but this is the general consensus of what I'm going to be talking about today. Okay, so I'm going to go through each one of the lectures individually. And on the bottom right corner, I have listed the textbook pages where you can find it for that particular lecture, where that material is. All right, so we're going to start with the basic organization of the nervous system. So ideally, what you need to know, I included some things here on the right that will help you remember what each one of the different components of the nervous system are for, or how to remember them. So things like the afferent and efferent neurons, make sure you get that down in your head. And when you have the exam in front of you, to just like you might look at it at first glance and be like, oh, I know it's this, I know it's that, but just go back, because those like little things are the things that get you on exams. Just take the time to reread the question and remember, you know, efferent is like escaping the brain or the central nervous system. That's how I remember the E in that sense. Helps me remember that efferent is going away, like it's exiting or escaping the brain and spinal cord, whereas afferent is going towards the brain and spinal cord. Additionally, somatic is for the movement, and then the autonomic, I like to think like it's like automatic. These are things that are out of your control per se. They just function in the background, like fight or flight. Let's say, for example, you're scared. You can't control that you're scared. You just get scared, right? Or you can't control that you're nervous giving a midterm review session for 100 people, but it happens. So, you know, you get a little stressed. And then whereas if you're having a good meal and you're just relaxing, you know, you feel the effects of the parasympathetic nervous system, which we know as rest and digest. So little things like that are things you want to be able to differentiate. Okay. I also have the enteric nervous system on there as well. I don't think it was mentioned in the course, but it's another nervous system that you might see later down the line. But it involves digestion and the gustatory system. Okay, so this is generally the what do you call it? The neurons, and I'm going to also talk about glia as well. So here I have a schematic that includes all the general parts of the neurons, and it's basically their whole job is to transmit signals to either nerve, muscle, or the endocrine cells. And primarily what you need to know is where the signals originate from, how they propagate, and then how this signal is sent from one neuron to the next neuron. And then know things like presynaptic versus postsynaptic. Know that the synapse is the gap between the neurons. Be mindful of things like what the axons are for and what the dendrites are for. Like just know the different components and what they're for. You know, give yourself the time to review these things. Look at a schematic like this and know what each one of the structures are for. Now we're going to look at the glia. We have the oligodendrocytes, the microglia, the astrocytes, and the Schwann cells were discussed in the course. Ependymal cells were not discussed. At least I didn't hear them in the lecture, so you can correct me if I'm wrong. But I also did hear about the stem cells. So let's see here. So these are the general functions that they have. Essentially, what we used to know about in neuroscience was the glia cell were like the structure, or they held things together. And now we've begun to understand that they have a vast majority of functions, like they supply nutrients, they help insulate the neurons, and then things like the microglia are kind of like the immune system of the central nervous system. So if you find microglia in the brain, you know, their job is basically to clean up from pathogens or debris in the brain. So they provide like a support to the central nervous system. So here's what we have, a meme for you guys. Keep you guys, I'm gonna have several memes sprinkled throughout the presentations and keep you guys entertained. I know this material can be a little dry. So here's the microglia cells. Like I discussed, they're the immune system basically of the central nervous system. Their role is to pretty much get rid of bad guys in the brain or bad things. And we've got the Schwann cells. These are the cells that produce the myelin sheath, and then they are primarily in the peripheral nervous system. We've got the astrocytes. They're the ones that provide like, they have the capillaries and they provide nutrients to the cells. They provide what they need as sustenance to keep them alive for the neurons. And then we've got the oligodendrocytes. They're essentially like the Schwann cells, but of the central nervous system. So they also provide the myelin sheath. So just be aware of those differences, because sometimes professors like to throw like a trick question, and if you're not sure of which one belongs where, then you can mix that up. Here we talk about chemical disequilibrium, and throughout every physio exam and every textbook I've seen, this is actually like usually something that you're going to get some kind of question on. I remember when I took my first midterm for physio last year, they had some general consensus, like a question that kind of gauges to see if you understand what belongs where. So just know things like where the high concentration of each one is at. So know that sodium is higher outside the cell, whereas potassium is higher inside the cell. You don't have to know the exact numbers. You would just have to know where they're more concentrated, because sometimes they like to give scenario questions and try to see if you understand where things belong, either in or outside of the cell. Okay, this might not be accurate, so you can refer to your textbook if you want a more accurate measurement. And then this is actually the graph that he used during the lecture. So this kind of gives you a more visual representation of the concentrations of the individual ions within the cell and without the cell. So here's a tip. Yeah, just be clear that you know potassium concentration is greater inside the cell and sodium concentration is greater outside the cell. That's going to help you in physio, neurobio, and a bunch of other courses depending on what you're taking, human bio. But all throughout bio, you see this constant pattern. So just always remember that. So how I remember it is, what are bananas famous for being rich in? And anybody can unmute themselves or just drop it on the chat.
Potassium.
Great job. Okay, gold star. Yes, bananas are famous for potassium. Okay. So want to hear a joke about potassium? K. Get it? K plus. Anyways, sorry, a little cheesy. I know. But then there's things that you also have to consider, like sodium. Sodium is more highly concentrated outside of the cell, right? So if anybody wants to like guess what is, or they can include their own opinion on why is it not good to take a lot of salt. What could potentially a lot of salt do to the body?
Then water will go out.
Exactly. So if water follows the concentration of the salt, right? So if the water leaves, if water gets drawn out of the cell, then it builds up in the extracellular fluid. And that's what essentially causes high blood pressure. So when people have high blood pressure, they're told to restrict or limit their intake of sodium. And basically how I remember which one is in and which one is out, you wouldn't mind eating a lot of bananas, but you would not want to eat a lot of salt. So I would take in banana and leave the salt out. I don't know if that helps you, but it's a little cheesy, I know. But little things like this help me remember, things down the road, so you don't have to think those through later. All right, so the Nernst equation and the Goldman equation are both equations that were mentioned in the lectures. You don't have to memorize every individual component, just know how they play a general role or what they're meant to describe. I am going to have a question for all of you a little later to test your knowledge, to see how comfortable you are with the equation. And it's actually a question that I received on an exam, so it's hopefully going to be helpful to you. But this just kind of breaks down how we derive the Nernst equation. And then this is the Goldman equation. And the difference between the two, essentially the Goldman equation focuses more on the reversal potential, whereas the Nernst equation gives you the membrane potential of the cell for that one particular ion. And the Goldman equation gives you the whole list of ions, like you're looking at each one individually and then adding them all together. So here we have the phases of the action potential. Know this chart. I saw this on my exam. They love to quiz on this. They love to ask questions like, when is this channel open, or when is this channel inactive? Which ions are flowing in and out. So just know each one of the steps and know which ion is basically taking priority here or what it's doing. Okay. So like, for example, at the resting, we're at the cell's resting membrane potential, which is below threshold. That's important. Know where threshold is and know what happens once you reach threshold. So at this point, sodium channels are closed. Potassium leak channels are the only thing that's open. That could be a trick question. They might say something like sodium channels are closed and potassium channels are open. They are not. Sodium channels are closed, potassium channels are closed as well. There is a subset of potassium channels which are referred to as leak channels, and these enable potassium, very minute amounts, to flow through the cell, and that's what maintains the resting membrane potential. So when we get to step 2, this is once a depolarization event has occurred, it reaches a threshold, and then this triggers the sodium voltage gated channels to open, and this causes a sudden influx of sodium, and that influx further depolarizes the cell. Depolarization meaning it's getting more positive, so it's getting away from that threshold value and climbing to closer to 0, let's call it. Okay. At this point, when we reach overshoot, so let's say that the sodium channels were open, and they were open for such a long time that the cell's overall potential went to a more positive value than intended. It overshot it. But then the sodium gates begin to close and more of the potassium channels start to open. And now if we know that sodium concentration is higher outside the cell, and now it's closed so it can't come in, now the potassium channels open, meaning potassium can go from inside the cell to outside the cell. That's going to carry positive charge from inside the cell and cause it to leave the cell. And that's why you see that downward trend from steps 3 to 4. And that's why when the potassium channels are open, the positive charge is leaving the cell and it's causing it to become more negative. And efflux is the opposite of influx. So know that, because it could be on a test question. They might just say efflux and expect you to know what efflux means. Efflux is exiting, okay? And lastly, we have the recovery step. And at this stage, the potassium channels were open for an extended period of time, and that positive charge left the cell and an excess left, and it made it more negative than intended. But your neuron overall wants to be at the resting membrane potential. So now that once you reach that point, the potassium channels go ahead and close. Sodium channels also close, and then this causes the cell to reach a resting membrane potential, which is the desired goal here. So essentially, that's how I feel every time I learn this. Every professor, and every time I learn it again, I have to every time get a little bit confused. So take the time to digest these steps and just quiz yourself, leave a little blank and know which cells are going in, which cells are going out. I mean, which ions are going in and which ions are going out at each one of the steps. I think that's the most beneficial here. So I don't know if you wanted to take this, Ariella, since you made these, but if not, I can try.
Ariella, you're on mute. Can you unmute yourself?
Oh, sorry, thanks for the catch. So I made another table to help differentiate between graded versus action potentials. So Jose, if you could help me uncover what's underneath. Beautiful. So graded potentials, they occur at the dendrite, cell body, and at sensory receptors. So they're relatively weak, but it depends on the stimulus strength, how strong or how weak they will be. And the direction of change can be depolarizing or hyperpolarizing, depending on the stimulus again. And the summation can be spatial and temporal. So for graded potentials, a key thing that's different is that it has no refractory periods, and the channel types can be ligand signaled or mechanically signaled. And the ions that are usually involved in this type of potential are sodium, chloride, and potassium. All right, and for action potentials, so these only occur on the axon, and it's either all or nothing. So the direction is going to be depolarizing, so it only goes in one direction, and there is no summation. Once there's an action potential, like I said before, it's all or nothing. And it has two types of refractory periods, which I will explain more depth later on. There's absolute and relative. And it only has a gated channel that is signaled by voltage. So the ions that are involved are sodium and potassium.
All right, so this is another overview, basically, of what Ariella discussed. It's essentially the same thing, just in different words. So maybe you'd want to go back and look back at this on your own time, but it's the same thing, just to repeat, to just reiterate the point and drive it home. And here we have graded potentials, and he showed in the lecture something similar to this, and how as the depolarization spreads out across the axon, you see that certain regions are depolarized while the other regions are not. And you want to focus on how this charge on a graded potential, it kind of loses strength as it proceeds down the axon. As it travels down the axon, it doesn't have the same like driving force while it's making its way down. So graded potentials they can easily get lost along the way. It's like, think about it like you're trying to swim through jello. It becomes tougher to make your way all the way across. Whereas the difference here would be an action potential. Once it's fired, it makes its way all the way across. And then here we have that generic chart that they show, and they like to show things like this. Basically gives you a nice grasp of the overview and difference between graded potentials and action potentials. So here, graded potentials can be hyperpolarizing or depolarizing. You see where like 1, 2, and 3, you can see it has a downward pattern. So these would be considered hyperpolarizing, where 4 and 5 are depolarizing events. And then it isn't until they reach a certain threshold where, for example, 6, 7, and 8, where this is where an action potential is actually triggered. And that's the difference between the two, essentially, because you can have graded potentials that are added together, and this effect could be a plus minus and never really reach threshold and not propagate the signal downward. Whereas action potentials, once you reach threshold, that's when the action potential takes over, and it's only in the forward direction. It only propagates the signal. Graded potentials can lose out. Yes.
Sorry, if you don't mind. So there's two questions. One is asking about this slide. So Andrew is asking, with graded potentials, when they get weaker, is that more negative or more positive, or does that depend on if it's a hyperpolarizing or depolarizing event?
Good question. So a graded potential would be like, let's see, like you made a mistake, right? Like you met a girl or you did something and you made a mistake, right? And over time, you're hoping that she forgets about this mistake, okay? Whereas maybe you did something good and then over time, she might forget. Now the action potential would be like, what you just proposed, and now she's never gonna forget that because you're ready to commit, right? I'm sorry for the analogy, I couldn't think of a better one, but graded potentials have an additive effect, and it could be adding a plus or adding a minus, and over time, well, essentially on an axon, over distance, they kind of lose strength. They kind of get forgotten, I'd say. Does that help? Or did it make it more confusing? I can't see the chat. Anybody want to speak up?
Andrew, does that help? Oh, yes. He says he gets it now.
Okay. And then one that I remember, and it kind of helps me, is the whole graded component. I break that down and think about it like 1, 2, 3, 4, 5 are like your grades, like over the course of a quarter. Not saying that you're a bad student, but let's just say you get a bunch of bad grades and a bunch of good grades. Until you reach threshold would be like you passed the class. So even though you did bad on the first quiz, you did bad on the second, eventually towards the 4th and 5th, you start getting some momentum and you have good scores. Hopefully you reach threshold, hopefully you pass the class and you move on to the next course. I don't know if that helps, but an action potential is essentially you passed and that momentum drives you forward. There's no going back once you pass the class, but graded potentials can have plus and minus effects. I hope that these analogies help. I don't know, I like analogies. Ariella?
All right, so moving on to refractory periods. So like I said before, there's two types, absolute and relative. So first off, absolute is when the sodium channels are inactive. So that means you can't have an action potential at this time or another one. The excitability is 0. And for the relative refractory period, at this stage, the the sodium channels are closed. So as you see in the graph, the excitability is increasing. So the local current can trigger another potential during this time. And another thing to mention is these periods allow the action potential to move in only one direction, which is why it's different from graded potentials and how graded potentials are bidirectional. So I have a clicker question for you to test if you heard what I said in the past slide. So during absolute refractory period, a neuron cannot generate another action potential because A, the sodium channels are closed and excitability is increasing. B, sodium channels are closed and excitability is 0. C, sodium channels are inactive and excitability is increasing. Or D, sodium channels are inactive and excitability is 0. Beautiful. Looks like you guys got it. The answer is D. So during the absolute refractory period, the channels are inactive and excitability is 0. So local current flow or a graded potential can't trigger an action potential during this time. All right, let's move on to propagation rates. So these are just factors that can increase the rate of action potential, such as resistance, the diameter of the axon, and myelination. So a larger diameter leads to less resistance in the axon, and myelination is just insulation, which makes less leakage of current flow. And next up, we got saltatory conduction. So this just pretty much means jumping across nodes of the Ranvier. So as you can see, there is the myelin sheath, the ones that have the beige circle. Yeah, beige, let's go with that. And that's what minimizes the current loss. And the nodes is when it doesn't have, it's like just the blue tubes, and they contain gated channels, which trigger the production of graded potentials in the myelinated region.
All right, I guess I'm back. Yeah, so again, if you want to take time to look over the slides at your own time and your own pace, this would be a general overview so we can see the things that we discussed. And here you see a picture of a neuron. It's a cartoon photo, but it kind of gives the same general idea of what we've been talking about this whole time. So these hands that are reaching out are the dendrites, and then the axon is that next to that arrow where it shows one way, and it's that whole long body that eventually reaches the axon terminals and it passes the message along to the next neuron. So that's the general consensus of like the entire neuron and how it functions. So we're going to talk about the difference between temporal summation and spatial summation. So here I made this for you guys, by the way. Here we have two leads and we're going to place them at different parts of the neuron. This is the postsynaptic neuron. And each one of those leads are going to pick up a recording. This is actually something that we do in neuroscience a lot in labs. So it's good to get exposed to it early and kind of understand the basic component of how this works. So this first neuron on the left is the presynaptic neuron. And the neuron on the right is the postsynaptic neuron. And that space or that gap in the middle is the synapse, okay? So what we're going to do is we're going to send a signal starting from the left and make its way down to the right. And in temporal summation, you're sending a signal from the same presynaptic neuron. It crosses the synapse, well, it makes its way down the axon, it crosses the synapse, and then here at the region of the dendrites, we have a slight peak, if you can see that, and then down at the axon hillock, it's an additional slight peak. However, this peak that you see is not strong enough to cross the threshold and cause the postsynaptic neuron to fire. So then you have to send another one in a short window of time, as you see it propagating down the presynaptic neuron, across the synapse, and this time it causes or generates a bigger peak, which makes its way down to the axon hillock. And since it was in such a short window of time, it kind of adds up with the previous one. The previous one did not fade away, it didn't have time to go away. It still was kind of there. There's like a residual charge there. And it causes it to add up to eventually cross threshold and fire an action potential. And this action potential is now fired from the postsynaptic neuron, and it makes its way down until eventually you've got a happy neuron. Okay. Now, the difference between that and spatial summation is time. Okay. Temporal, think time. I don't know a good analogy, but just know that, you know, what do you call it? Shrimp tempura. It's about that time, right? I don't know if that helps, but yeah. So now we're gonna look at spatial summation. Let's see. I don't know why it's not doing anything. Okay, so spatial summation refers to neurons close to one another. So where temporal was one neuron firing a signal in a short window of time, spatial summation could be multiple neurons that are close to each other and firing a signal to the postsynaptic neuron. So these two presynaptic neurons are going to fire their signals down their axons to reach the postsynaptic neuron. And again, we're going to take a measurement from the leads on the postsynaptic neuron, but this time we're going to ignore the top box. See, there's no lead plugged in there, okay? We're only going to focus on the bottom box. So these two are going to send a signal together, and they're both going to cross the synapse, and they're going to summate and be able to be strong enough to reach threshold and fire an action potential. So you're going to see that this postsynaptic neuron is going to fire an action potential. You see, and then we have another happy neuron. So the difference between the two is temporal involves one, it involves a short window of time, where spatial involves neurons in close proximity to one another or close in space to one another. Okay, and their effects can be additive. Now, I have a question for y'all. Let's see who can answer this one. First one gets a gold star. So these purple neurons are excitatory presynaptic neurons. This red neuron I introduced here is an inhibitory presynaptic neuron. If these were to fire, if you had, let's just say, some level of plus and some level of minus, what kind of effect would you see? What would happen? What could potentially happen here? Anybody can unmute their mic and feel free to answer this question. If you're still there, I don't even know if I'm still here.
Wouldn't the pink one be a depolarization and the other one would be a hyperpolarization? And then one, no?
I think
I know one cancels out the other.
Okay, so you're on the right track. One cancels out the other, but you said the pink one. Are you referring to the bottom one?
Yeah.
Okay, so the pink one, let's say the bottom one, if it's actually negative and like let's just say the signal that it's sending is an inhibitory signal. It's not an excitatory signal.
Oh.
It wouldn't depolarize. It would hyperpolarize. Okay, so if this bottom one hyperpolarizes and the other two depolarize, so you're going to have an effect that, yes, it kind of cancels out. So this is what we would see here. If you look at our reading on the lead there that I took with my fancy measurements. So we would have like a canceling out effect. So that's the key with like spatial summation. You might get asked a question in regards to like what would happen if you had a plus and minus effect. Like if you had excitatory and inhibitory neurons, you would see a canceling out. All right. Did anybody have any questions on this slide before I proceed? Because I know it was a lot to take in.
Yes, I have a question about this.
Yes.
So are we saying that an action potential doesn't occur?
In this case, no, because as you see, it doesn't reach threshold. So you would see a peak like this if it reached threshold. In this particular case that I provided for you, the inhibitory neuron was powerful enough to inhibit the excitatory neurons. However, there are cases where you can have, there could be a multitude of presynaptic neurons, and their effects might be strong enough to overcome that inhibitory effect and therefore propagate an action potential. So it really is scenario or case by case that you have to take this. There's no like definitive answer, but in this scenario that I provided for you, it did not fire an action potential. It did not reach threshold.
And that's because of this almost like graph image that you have right here.
Yes, so you would see.
I don't think I noticed it the first time. Okay.
Oh, okay. Yeah, I know. It's kind of quick. Sorry about that.
Okay, thanks.
Okay, anybody else have
few
any questions?
Just dropped in the chat. Okay,
I can't
so
see the
Andrew, that's okay. I'll moderate. So Andrew is asking, in the actual depolarization / hyperpolarization event, how can we differentiate an inhibitory signal or an excitatory signal? Do they both still have an influx of sodium?
Oh, okay. So I'm actually going to get to that in a couple slides, but just to give you a preview, different ions have different effects, and there's different concentration of those different ions inside and outside the cell. So one example that they like to throw at you is chloride. So chloride is, if chloride was to flow into the cell, chloride brings in a negative charge. And if it brought in a negative charge into the cell would not depolarize. It would not become more positive. Instead, it would become more negative. And things you have to consider the different ions. You have to, it's really a case by case. So when you're depolarizing, positive charge would essentially be coming into the cell, like sodium. Now, if you're hyperpolarizing, you can have the effect of potassium leaving the cell, which is a plus charge leaving the cell, making it more negative, which would be hyperpolarization. Or you can have something like chloride, a negative ion, entering the cell, bringing more negative charge into the cell and bringing it further away from threshold, also hyperpolarizing. Does that make sense?
Okay, and then, yes, he said that it does. And then Brandon is asking, do different gates open depending on if it's excited or on if it's an excitatory neuron or an inhibitory neuron?
Do different gates open? Okay, so the neuron doesn't just have one particular channel. Like these purple neurons I showed don't just contain sodium. They can have different ion channels, right? And these ion channels are triggered by the overall charge of the neuron. So if the neuron has, if this purple neuron has a bunch of sodium channels and they get triggered because they're voltage gated and a charge is coming down, it would open those channels and allow for influx of sodium. However, each neuron is different. There's no like, every neuron has 10 sodium channels and 5 potassium. Every neuron is different and has its own certain amount of each of the ion channels. And they're triggered by things like voltage, or some of them are triggered by ligands. So something binds and it opens the channel to allow the ions to flow through. So each neuron is different. Each neuron contains a multitude of channels, and they're all activated by different mechanisms. You know, there's metabotropic, ionotropic. There's different ways of opening these channels. So there's no such thing as just like knowing what each neuron has and all that it has. You have to kind of look at it like big picture. Like they will tell you it's an excitatory neuron, and that means that the signal that it fires is overall going to bring it closer to depolarization. Whereas like an inhibitory neuron would do the opposite. You don't have to know the specifics and the mechanisms. You just have to kind of understand the general idea. What we've discussed should be good enough for the exam. If you want to go in depth, like there's like the neuroscience courses that do kind of give you like a more serious breakdown, but I don't think you need to worry about that right here.
Mm-hmm. And I've been trying to moderate the chat as much as I can, but if I didn't get to your question, you guys can feel free to ask us after we're done with the presentation.
Definitely. So here's the types of synapses. Essentially, you need to know electrical versus chemical. And in particular, the electrical have these gap junctions, means it's kind of like a tunnel that they share with the adjacent neuron. And that tunnel is formed by these connections, these proteins that form this pore and allow the charge to flow through. And since they're pretty much touching and they have a tunnel that they can share charge through, it's pretty quick. And this charge can pass from one neuron to the next. And you tend to see these kind of things a lot in like cardiac tissue and some smooth muscle. And it just makes it so that way the overall charge just easily flows through these gap junctions. And it is able to excite the postsynaptic neuron. Whereas things like chemical synapses involve the use of neurotransmitters. And these neurotransmitters would therefore trigger the ion channels on the postsynaptic neuron to open. So like a neurotransmitter would be like a lock. They like to use this lock and key analogy. So I'm sorry, the neurotransmitter is like a key, and it'll go and touch the ion channel of the postsynaptic neuron and open that lock, and therefore allow ions to flow through it. So here we've got the chemical synapse. You just need to know the relevant parts and functions here. Just take a look at these, know what they are. Sometimes they like to have a little schematic, like a black and white image of a neuron, and then ask you to label certain parts or what would happen at different stages. And this is just a general overview. Does anybody have any questions up until this point?
I believe Catherine Catherine is asking, what's the difference between connexins and connexons?
That's a good question. I don't know if there is a difference. Why? Where's this coming from? I don't know.
So connexins is the protein that forms the structure, and then the connexon is like it's not the protein, but it's like the chemistry of like how the proteins come together.
I see online it says connexons are formed by connexins. So I guess the connexins are the individual subunits that make that pore, and the connexon would be referring to that whole pore. So I hope that helps.
Then, okay, and then Dundon says connexons are made of connexins.
Hey, thank you, Dundon.
Erica is saying, so should we say that the signal is inhibitory / excitatory, or the neuron itself is, in the case that the neuron has the available channels for both?
That's a good question. So the neuron itself, it depends on its function. So you would label the neuron based on what it's doing. So if the neuron is sending an inhibitory signal because of the particular situation that it's in, let's just say it's in some solution that enables it to want to go ahead and open its sodium channels and therefore make it excitatory, then it's an excitatory neuron. But if it's that same neuron is in a different solution and therefore causing, chloride to flow in, and then the signal that it transmits, it's hyperpolarizing, it's an inhibitory neuron. So it really depends on the situation that the neuron is in. You can't just label every neuron by what it looks like. You got to label it by what it does. Just like people, right?
Yeah, so it does depend on the signal. There are cases where like you'll have, for example, GABAergic neurons. So these will mostly respond to inhibitory signals. But I think it does depend on the depolarizing signal. So like once the depolarization occurs and the neuron senses it, then an action, it'll be an excitatory signal. Depolarizations are positive, so they're excitatory.
We're good? Anybody else want to ask a question? Cool. All right, so here we need to know the events and how this forms exocytosis. This is a big one to know. This is the whole basis of how the neurons communicate, how they send the whole chemical signaling, and how they activate one another. I know that Monica is going to discuss some of this in muscles a little later, but just to give you a general overview, the action potential makes its way down, and then it reaches the axon terminal. This causes a depolarization, and these channels that you see here, these calcium channels, are voltage gated. So now that this neuron has been depolarized, it allows these voltage gated channels to open, and calcium likes to flow in. And when calcium flows in, there's a whole mechanism behind this, but basically the calcium helps facilitate these synaptic vesicles to make their way down to the terminal and bind to the terminal and essentially open up and release their contents. And when they release their contents, these neurotransmitters make their way down to the postsynaptic neuron, and then they bind to the specific ligand receptors on this postsynaptic neuron. And here we have the recycling of these and how we make them essentially. So in our bodies, we use acetylcholine a lot for a lot of different things. And acetylcholine is made up of acetyl-CoA and choline. And there's an enzyme that we have that hangs out. It likes to hang out in the synapse. And what it does is when this acetylcholine is in these vesicles and eventually released, it makes its way down and it binds to these receptors in the postsynaptic neuron. And then that acetylcholine is hanging out in the synapse where it meets the acetylcholinesterase, which breaks it down into its individual components again. So what you see in 2, that blue ball thing, the blue oval, that right there is the enzyme. And that's what's doing the breaking down of the acetylcholine into acetyl-CoA and choline. And what happens is this choline gets transported with sodium and it gets transported back into the presynaptic neuron. And the way that this gets transported is a co-transport mechanism, meaning that both of these are going to flow together across the membrane and back into this presynaptic neuron. However, the concentration gradient for sodium is high on the outside and low on the inside. So it wants to move in. And then the choline hitches along for the ride. It's as if sodium paid the toll and brought choline with it. Okay, and it brought the choline with it against its concentration gradient because choline concentration is greater inside of the neuron. And now that choline is back in the cell, it can be reused or recycled to make more acetylcholine. And then here we have the difference between ionotropic and metabotropic synapses. So we know that ionotropic are much faster, and the way that they work is kind of like a lock and key situation where they're ligand gated. And what happens is once the neurotransmitter binds, then it opens the pore or channel, and then it enables the ions to flow through. Whereas metabotropic refers to, it's like an intracellular, an extracellular event occurs, and then it causes this whole signaling cascade to become activated. And as you can see, this pink would be, for example, G protein complex, and a neurotransmitter comes in and binds to it, and then this G protein complex enables these blue blobs here. They're the whole intracellular signaling cascade, which eventually activate a channel or pore and enable the ions to flow through. So these are much slower. And the most common example is the G protein coupled receptors. And you're going to see these throughout physiology, biochem, molecular bio. So I made this for y'all. It took me a long time. It's essentially the same diagram that Professor Azizi has on one of the slides, but I guess it looks a little more, I don't know what you call it, retro, hopefully a little cleaner. It gives you the same general idea, but you can kind of follow how the neurotransmitter either binds on the left to the ion gated or on the right to the metabotropic receptors here. And if you bind, let's say, to the left, the ion channels can open, and depending on which ions flow through it, like for example, sodium, which would be excitatory, this would cause an excitatory postsynaptic potential. Or if it opened an ion channel where potassium were to flow out or chloride were to flow in, like we discussed earlier, then this would be an inhibitory postsynaptic potential. Essentially, if these ions closed, then like the opposite would happen. Then you can have things like the sodium that isn't able to enter the cell, and that could be inhibitory. Or let's just say, for example, potassium could would be able to leave the cell through leak channels or other forms, but it could become excitatory if the signal overall, if the cell overall is positive. And then the second messenger systems is what we've talked about, and this is a whole cascade that can affect protein synthesis and other coordinated intracellular responses.
Can I have a question for the previous slide? Okay, so for the left one, is that the ionotropic, and the right one is the metabotropic?
Yes, exactly.
Okay, and also for the, so I kind of like differentiate these types. So for ionotropic, the ion channel can only open, but then for the metabotropic, the ion channel can be open or closed?
Yes, and that's dependent on whatever binds to that metabotropic receptor. It starts off the whole cascade. It could have different effects. It could be open or closed, and then it can lead to other effects. But metabotropic could have excitatory or inhibitory effects overall. It's not defined by one.
Okay, thank you.
Any additional questions? Okay. He talked about these very briefly towards the end of lecture or nerves 2. And I don't think you're going to be quizzed on them, but just kind of know the general idea. I just included this here for y'all, but I don't think I need to spend too much time on it. And then here we finally reached nerves 3, so almost there. Let's see, hang in there. I'll try to keep you entertained towards the end. Oh yeah, the brain is the most important organ you have, according to the brain, of course. Let's see, so CNS architecture, he talked about brain, spinal cord, and you know that that's the central nervous system. I don't think you need to know the different sections, but just know that there are different sections and general idea of where they are. But each of these have nerves that leave the spinal cord and go out to the periphery. And that's basically the point from central nervous system to periphery nervous system. That's the intersection for them. Left and right hemisphere, and the cerebellum, which is involved in movement coordination. So then here we have the frontal, parietal, occipital, and temporal lobe. I say know these because you're going to see them either in this class or in so many other classes, med school or wherever you end up. This is something that they like to just kind of see if you understand this just for basic knowledge. So it could be a very easy test question. It could basically be a freebie. And these would be each one of their functions. If you want to go over them, or if you have questions, you can go ahead and ask, but if not, I'm going to proceed so I don't spend too much time. And then here we get to gray and white matter. Know that they're inverse from brain and spinal cord. So what you see on the outer regions of the brain are gray matter, and on the inner you see the white matter, where in the spinal cord, the opposite is true. So sometimes they like to trick you with that one. We know that the white consists of the myelinated axons. It's white because it's fatty in nature, and that's actually what gives it its color. And when we stain the brain, we do see the difference from gray matter to white matter. And the gray matter it contains a lot of cell bodies, and it also has the dendrites and axon terminals of different neurons. So it gives it its nice rich color. And that's essentially where a lot of the communication occurs, whereas the white matter is where the transmission of that signal occurs. And then when it reaches these dendrites or axons of different neurons is where the actual activity takes place. Let's think of it. And white matter is where it's sent out to the next neuron before it performs its function. So these are the major brain regions. I included a bunch of extras, but you don't really need to know all of them. So I'm going to go ahead and highlight the ones you do need to know. Know the thalamus. I think of that as a relay center. It receives something and it sends something up to its specific region of the brain. So like, for example, if you get like an auditory or let's just say like a nice smell or a nice sound that you hear, the thalamus receives the signal. It's like a relay runner, and then it sends it up to its specific brain area where it'll go ahead and process it. So if you saw something interesting, the thalamus gets it and it passes it on to the occipital lobe or the visual cortex. And then we've got the medulla. This controls the involuntary functions, all of the basic things that you don't even think about that are keeping you alive. Oftentimes in movies, you see where people go and they snap the neck. That's because they're snapping the medulla. The medulla is an ancient brain region that we've had, if you believe in evolution. It's been around for a long time. All of the most basic animals have it, and it controls things from breathing to heart rate, and it's keeping us alive. And then we've got the pons. This actually controls like your rate of breathing, or it controls certain aspects of basically the mechanics of your breathing. Whereas the medulla is like the on and off switch. Either you're breathing or you're not. The pons dictates how you breathe, essentially. And then the pituitary, you're going to see this a lot. It's involved in hormone secretion, and its role overall, it's got a bunch of roles, but it's primarily influenced by the hypothalamus. And you're going to see the HPA axis, which gives it's basically like the boss telling the pituitary, the employee, what to do. And it goes ahead and sends all of its hormones out to the body, which is involved in a multitude of effects, from reproduction to growth to all kinds of things that keep us becoming the young adults that we've become today. So here we're going to focus on the hypothalamus. You spent some time on this. Know generally what the hypothalamus is for. These are all its major functions, like hormone release, because it's related to the pituitary. It also regulates your body temp, daily physiological cycles. This is like your sleep and wake cycles, just the things that your body does routinely, oftentimes. It regulates your blood pressure. If you're hungry or thirsty, you can blame your hypothalamus right now. I'm going to go ahead and skip the next one, but just be mindful that you practice safe practices when you're engaging in that kind of behavior. Sleep and wakefulness, again, like that kind of has to play with your daily physiological cycles. It's part of that whole system. And then lastly, people usually associate your emotional responses with the amygdala, but the hypothalamus also plays a role in your emotional response, because your emotional responses are also somewhat physical. And then here I've listed different hormones that are where the hypothalamus has an influence on the pituitary, and in each one of these hormones plays a role in your overall body production, different regions of your body, how you grow, reproductive behaviors. So they work together in conjunction. So the hypothalamus receives a signal and it sends it to the pituitary to say, hey, this is what you need to do with this information. Now we've got
a quick question about the previous slide.
Yes.
So should we remember? So should I remember like of the hormone that the pituitary secret, or just like kind of like
Okay,
familiar
so
with this?
for what he's covered so far, I wouldn't say that you need to focus on the reproductive hormones, especially because they're going to go in depth in the last unit of physiology. So in the last unit, they're going to focus on things like prolactin, oxytocin, more so, also follicle stimulating hormone, which I'm not seeing on here. But the reproductive ones, I would say focus on those. Just know that they're reproductive, but you don't have to know their exact roles until the last unit of physio. For now, focus on things like growth hormone, focus on, let's see what, thyroid stimulating hormone and adrenocorticotropic hormone. So focus on those more so than the reproductive ones. Now we've got the autonomic division. Like I mentioned before, autonomic is pretty much like, think of it as automatic. This kind of things happen, they're out of your control. Unless you're some kind of yogi or well versed in the meditation. It's difficult to control, you know, let's say saliva production or things like constricting your pupils. These kind of things happen on their own. And this controls like smooth and cardiac muscles. So your digestion, how your heart rate is working. And if you get excited or if you're at a relaxed state, these kind of things are happening in the background. So think of your rest and digest functions and your fight or flight. So finally, we reach spinal cord anatomy. I like to remember that the dorsal horns are on the back, just like a dolphin has a dorsal fin and it's on its back. And this contains the cell bodies of sensory neurons. So just think of sensing a dolphin. Okay, if you're in the ocean or near an ocean and you see a dolphin, you're going to get excited. You're going to sense that dolphin. However, the ventral horns are located in the front of the spinal cord, and these contain the motor neurons. And I guess you always want to run forward. You don't want to run backward. So your ventral is in the front and it involves movement. So forward, movement, front, and then dorsal is in the back, sensing. Okay, so know that difference because they like to play with the pathways a little bit to try to trick you. Like they like to throw in two-part questions to see, you might agree with the dorsal horn part, but you might mix up that it's motor versus sensory. So just differentiate those in your mind.
Jose, would you really quickly go to the last slide and explain exo versus endocrine glands? Thank you.
Sure. So I don't think he's discussed it too much, but the exocrine glands, if I'm not mistaken, are going to be, if anybody wants to help me, any additional tutors can hop in, but the exocrine glands are the ones secreting a hormone that eventually affects another gland. And like, for example, let's see, let me try to think of one. So an endocrine gland would be the glands on top of the kidneys, right? The adrenal glands. And then an exocrine gland would be a gland that's releasing hormone that it reaches eventually that adrenal gland and tells the adrenal gland what to do. So exocrine is kind of like behind the scenes. It's kind of like, let's say like the hypothalamus, I think is considered an exocrine, whereas the endocrine would be like the adrenal gland. So the hypothalamus directs information to the endocrine gland. Does that make sense?
Perfect. Thank you. Yeah, that makes sense. Great.
Okay, no worries. All right, and then we make our way to ascending and descending tracks. So ascending is to the brain, descending is from the brain. So going up is ascending, going down is descending. And then here we have, I mean, it might be helpful to know kind of where they are, like where you see these colored regions, but I don't think he's going to throw in a picture and say, hey, what part is this? You know, like this, I don't think he would do that. And then we've got the spinal cord, which is known as the integration center. There's a common theme with sensory, integration, and response. Just if you want to practice your pathways for physio, I think it's helpful throughout all of physio to just know which ones sense information and then where it gets integrated and then where you see the response. So in this case, things like rapid reflexes, their function is to protect us from harm. I'm going to give you an example of which later. And then here we need to know that the integration occurs at the spinal cord. And we have these things called interneurons. And these interneurons are between sensory and motor neurons. So when you've got a stimulus happening, like at step 1 here, you get poked by a needle, then it eventually makes its way down the sensory neuron. You sense that pain. It makes its way to the interneuron in the spinal cord. And then this interneuron passes that information to the motor neuron. and then that motor neuron allows you to pull away from the pain, let's say. And just know, like I had mentioned before, your dorsal in the back of the spinal cord is how you sense it. So that's why the sensory neuron is going to the back of the spinal cord. And then it sends within that spinal cord, through the interneuron, it sends a signal to the front of the spinal cord. And you always want to move forward, so the ventral. And this is why it goes ahead and sends the signal down the motor neuron. Okay, integration is minimal at this stage, and don't forget that there is still a signal that is sent upward up to the ascending tracks up to the brain. So the brain does receive the signal that you felt pain, but usually you pull away before you even know to pull away. It's kind of it's a reflex essentially. So you touch something hot, you pull away really fast, and then you feel, oh, that was hot. Yeah, and this pathway involves 3 neurons. It's a rapid pathway. So here, the basic one that everybody uses as an example is hot stimulus, pull away. So you have your stimulus, your receptor, which is on your skin, afferent sensory neuron, which is the one we talked about, makes its way to the dorsal horn, the back of the spinal cord. The interneuron passes that to the front of the spinal cord, the ventral, known as the ventral horn. And then the efferent or motor neuron is leaving the spinal cord and making its way down to the effector, which eventually enables you to emit your response, which is pulling away. So this is the whole consensus behind the withdrawal reflex. Sometimes they like to mix these up and try to see if you can figure out the order or what belongs where. And lastly, we have these. I would say know each one of the receptors and the neurotransmitters that are involved. Take the time to just memorize them, separate them in your mind, which ones are for which. I kind of try to do a breakdown here so it can help you remember them. But yeah, we make our way to thermoregulation, which is the last thing he discussed in lecture 3. And this is the example of your body regulating your temperature. And essentially you do this through the hypothalamus. So you sense it in your skin. And then because you sense like something is, for example, cold, you sense it in your skin, you send that signal up to the hypothalamus sends it down, and it just sets off a whole cascade that begins from the detection of the temperature change all the way down until your body responds to it. And then you do things like, for example, you would dilate your blood vessels or you would constrict your blood vessels according to what kind of temperature you're feeling. So if you're cold, would your blood vessels on your skin surface, would they dilate or would they constrict? Anybody can throw the answer out. I don't know if anybody has answered, but I hope y'all got it right. So essentially, if you're cold, the blood vessels would go ahead and constrict. That'd be the response. So you'd want to pull the blood away from the skin surface and keep it more internal. So by constricting, you're not enabling the blood to flow easily through the outer skin surface. So that way it stays closer to the body and keeping it warm. And that's just one big example, but there's many that you can think of, and they kind of have opposing effects. So the whole basis behind homeostatic response is that it's a lot slower than that fast reflex response that we learned about. And the thing that you have to know is that you're trying to maintain a constant set point. You're trying to achieve homeostasis, so you have to constantly be receiving input, sensory input from outside of your body. And then what you do with that information, you integrate it and try to respond accordingly. So try to think of scenarios in your head of things of how you maintain balance and how your body would respond, because those are very common questions in physio. And lastly, I have some questions for you. Take the time to look at these and see if you can answer them. Feel free to do so on the chat. Let's start with number 1. So sodium-dependent neurotransmitter reuptake is? Anybody? Or anybody want to say it out loud? Anybody confident enough to share? Sodium-dependent neurotransmitter.
Is it none of the above?
Why do you think it's none of the above? What do you think it is?
Because like it's not, or maybe it's facilitated diffusion.
Facilitated diffusion, you think?
Because Na plus wants to go in, but NX taking along the neurotransmitter with it. So it doesn't require ATP, so it's not active.
Okay.
But I don't know if that counts as facilitated diffusion.
All right, so I'm gonna give you guys the answer and kind of explain it. So it doesn't require ATP, but it still requires energy. So the correct answer here would be secondary active transport. And that's because it uses the energy stored in the sodium concentration gradient to move that neurotransmitter inside the axon terminal. And that's the whole basis behind secondary active transport. It's dependent on something else to make that movement happen. And in this case, the neurotransmitter moves because it's dependent on the sodium concentration gradient. Does that make sense?
Can I have a question?
Of course.
Okay, so for like the sodium dependent neurotransmitter reuptake, are we talking about like the sodium and choline? Or just like a general?
Yes, that's one example, and that would also be called co-transport. But if you want to, like, let's say he didn't give you that answer. Like if this was your test question, secondary active transport is the most accurate answer here. Sometimes you might know things to fit one definition, but co-transport is like an umbrella term, which can also include things like secondary active transport.
Okay, thank you.
No problem. Okay, so next we have, sure, who's asking?
Okay. I was just wondering, and I think you already explained it, so sorry if I have to make you repeat, but can you differentiate between primary and secondary again?
Okay, primary involves the use of ATP. It's a direct need for energy to make this happen. It's a process that requires energy, which makes it primarily active, let's just say. So if you have that currency, you're able to make your way across or transport it across. Whereas secondary does not require ATP, it requires some other mechanism to make this movement happen. So because the sodium concentration gradient is causing this this whole mechanism here, it's dependent on the sodium concentration gradient, not ATP. So it's a secondary active transport. And because that sodium concentration gradient is there, it's enabling the neurotransmitter to be moved across the membrane.
Thank you.
Yeah. I have a question about this. So if this is dependent on a gradient, why is it active transport? I'm just a little bit confused by that.
Good question. In this case, I would say let's refer to Google. I mean, I did find this question in the textbook, and I tried my best to understand the answer. But if anybody has any like a better explanation, maybe they can provide that. But what exactly is your question again? If you can reword it.
My question was, so when you explained what the secondary active transport was, you said it doesn't require ATP and it uses a different mechanism of transporting the sodium using a concentration gradient.
Yeah, so apparently it's active because it uses energy, and active transport uses energy. So primary uses ATP, and then secondary uses the energy in the gradient.
Got it. And it's going against the concentration gradient, right? Still?
The neurotransmitter is. Sodium is going with its concentration gradient. So if it's a high region of sodium to a low region. So let's say, for example, you have high sodium outside the cell, and it wants to move inside the cell. And now something like choline hitches on for the ride. This can also be referred to as co-transport, but secondary active transport is another way of explaining co-transport, let's say. They're kind of like one and the same.
Got it. Oh, so just want to clarify, so co-transport is secondary active transport?
Yeah, essentially, yeah.
Okay, got it.
Okay, so the next question, I hope this is an easy one. Anybody want to just yell it out? I see.
That's hyperpolarization.
Yes, good job. Okay, why? Because it's becoming more negative. It's getting further away from threshold. And the last one, this one is a math question. I think I'm just going to kind of give it to you unless anybody knows the answer here. It looks daunting at first because they do kind of ask these questions but they make it very simplified. There's actually a trick here to make it so easy for you. Does anybody remember the Nernst equation, how to use it? Let's see, anybody have an answer?
Wait, are we expected to memorize the Nernst equation?
Not exactly, but I saw this question on my exam and I flipped because I was like, am I supposed to use the Nernst equation? And you kind of are. So here, I'm going to give it to you. So essentially, we know that I don't know if you can see this, but the ion concentration outside the cell goes on the top and the ion concentration inside goes on the bottom. So this is kind of like, you think it's a math-based question, we're going to need a calculator and everything, but you don't. Since it's 100 over 1, the log of 100 is 2. And then you get 61 times 2. So it actually is a very simple question if you understand the general way that Nernst equation works. So they're not expecting you to have that equation on hand and then apply it and everything. You just kind of have to know the concentration top and bottom and then knowing what to multiply by. I got this exact question and I'm going to be sad to admit that I got it wrong. I guessed it was like 110 or something. I forgot what it was, but I guessed wrong. They were kind of very similar answers. It was like 110, 120, and then there was like some ridiculous number. Like, so there was like 2 obviously wrong answers and then 2 answers that were very close. And then the whole trick was knowing that the log of something. So the log of 100 is 2.
So is RT over F equal to 61?
Right, yeah, so that's like the simplified version of the RT over F.
So then that means if it was like another, like say calcium 2+, then it would be 61 divided by 2 then, right?
Right, so the Z value is the valence number. The F is Faraday's constant, don't worry about that, it's like a big number. But that Z value would make it a +1 or +2, or, you know, depending on what it is. Like in how you said here, calcium would be a +2, and that would make it a +2 on the bottom, and that would be 61 divided by 2. And then if we had the same concentration, we would have whatever this value is by half, pretty much. Does that make sense?
Yeah, got it. Cool.
Yeah, so that's kind of like a question that they would ask. They wouldn't ask you to like it's not anything you need a calculator for. It's something you can figure out if you're paying attention to this general structure of the equation.
So we would just need to know like 61 times log of ion out for it.
Yeah, but the question wouldn't need a calculator. It would just be out over in. And then it's going to be something simple like this.
Okay.
Yeah, that's what they hit me with. And I was a little like, what? I didn't memorize this. You told me I didn't memorize it. But it wasn't about memorizing the equation per se. It's about understanding the equation.
Okay.
All right. Well, I hope this was helpful. You guys should take a look at that. I made this just for you. But yeah, this is it for me. This is my time. So I'm going to let Monica take over now. It was fun. Thank you all.
Thank you.
All right. So I realized that we are a bit short on time. I'm not trying my best to get through everything, but I am going to ask that if you guys have questions, try to save them till the end or just type them in the chat box. And maybe Ariella or Jose can help out answering. If not, my email is always available. Feel free to email me, just so that we make sure we get through all the content. Okay, Jose, please. Okay, so this is lecture 6. So we start off describing the 3 different types of muscle. So we have skeletal, cardiac, and smooth muscle. And all of these have very unique characteristics. So skeletal and cardiac are similar in that they're both striated, whereas smooth muscle is not. Both cardiac muscle and smooth muscle are union nucleated, whereas skeletal muscle is multi-nucleated. And then also the speed by which they undergo contraction is going to be fastest for skeletal muscle and slowest in smooth muscle. And then cardiac and smooth muscle are both regulated by the autonomic nervous system, so by your brain, whereas, or the involuntary parts of your brain, and then skeletal muscle is under voluntary control, so the somatic motor part of the division of your brain. So this is the inside of a muscle fiber. So we have mitochondria, which is our house of the cell, right? Where ATP is generated. It's going to be where we produce all our energy that's needed for contraction. The myofibril is the functional part of the cell. So that's the part where we're going to have the actual contraction happening. Sarcolemma is the membrane of the muscle. And then C-tubules are these kind of invaginations within the muscle that help propagate the signal into the deeper parts of the cell. And then the sarcoplasmic reticulum is what houses the calcium ions that eventually release out into the cytoplasm and initiate contraction. Okay, so this is just the clicker question. Which of the following functions does the T-tubule serve? Real quick, answer in the chat. Just take a few seconds. Okay, and then Jose, next slide. Yeah, so it is the allowing the depolarizing signal to rapidly reach the deeper regions of the muscle fiber. So sarcomere structure, we have the Z discs at the end, which kind of outline the borders of the sarcomere and they give the rigidity. The thin filament, which is also actin, and then the myosin is the thick filament, and then in between we have this titin, which is a structural protein, and it provides the elasticity that's needed for the actin filaments to go towards the M line during contraction. Okay, so real quick, I know Jose touched on this, but we're gonna talk a little bit about what happens at the neuromuscular junction. So the first step is the action potential propagates to the axon terminal, which is the end part of the axon, and the voltage gated calcium channels open, releasing an influx of calcium into the neuron. And when that happens, the vesicles fuse with the plasma membrane. And once they fuse with the plasma membrane, they're able to release all of the neurotransmitter that's inside, which in this case is acetylcholine. And then acetylcholine interacts with the receptor postsynaptically on the ligand-gated channels on what we call the motor end plate, which is the postsynaptic cell. And then the signal is then sent to the muscle using the T-tubules to trigger muscle contraction. And we'll talk a little bit more about how exactly that happens later on. Okay, another question. A certain drug was designed to block calcium entry into presynaptic vesicles filled with acetylcholine. What effect do you think this might have? Okay, next slide. Yeah, so see, a decrease in muscle contraction will be seen. Without the acetylcholine signal, the muscle won't contract. And of course, calcium is responsible for prompting that signal postsynaptically because it prompts vesicle fusion with the membrane. Okay, so excitation contraction coupling is basically when an electrical signal triggers calcium release by the sarcoplasmic reticulum, which is what we said. It's the motor end plate, which is the postsynaptic end of that receives the action potential. That's essentially what we're talking about when we say excitation contraction coupling. So the acetylcholine signal reaches the motor end plate. And then once it reaches that signal, after it binds with the ligand-gated channels, it'll travel down the T-tubule. And you'll see here in this diagram, the T-tubule is this big hole, this kind of big invagination, and it allows the signal to travel all the way down. And without the T-tubule, the signal might travel, but it won't be very fast. So the T-tubule allows for efficient reception of the excitatory signal, which is why skeletal muscle is able to achieve contraction relatively fast because of the T-tubules. And then the DHPR is basically a receptor that sits on the membrane on a sarcolemma, and it senses that excitatory signal. And then once it senses it, it causes a conformational change with the ryanodine channels. So the ryanodine channels are otherwise closed because they're bound to the DHPR. And so once the signal reaches down, they'll essentially shift apart a little bit from one another. And when that happens, that allows the calcium in the sarcoplasmic reticulum to release from the sarcoplasmic reticulum and into the cytoplasm. Okay, so a little bit more specifically, what happens once calcium is out into the cytoplasm? So calcium binds to troponin, and then troponin binds to tropomyosin. And then tropomyosin is this kind of orange band that you can see. And once tropomyosin binds, that exposes the myosin binding sites on the actin. So you guys see those, they kind of look like green olives. Those are the actin binding sites, the dark green part. So once tropomyosin shifts, that exposes the actin binding sites, or excuse me, the myosin binding sites on the actin. And then myosin binds to actin, which forms cross bridges. And that cross bridge formation is kind of the functional part of contraction, the ability to form cross bridges. Okay, so this schematic goes through the specific steps of muscle contraction. So myosin heads, they'll hydrolyze ATP using something called myosin ATPase. And that's an enzyme that essentially makes ADP and inorganic phosphate from ATP. Okay, and so when that happens, the myosin heads can then bind to actin, forming these cross bridges. And then the ADP is, in the meantime, still bound to the complex. Myosin cross bridges rotate toward the center of the sarcomere in order to form the cross bridge and perform the power stroke. And then as myosin heads bind ATP, the cross bridges detach from actin. So once we have ADP and phosphate, the power stroke is going on and the myosin is attached to actin. But once we add ATP back in, that causes detachment of the cross bridges. Okay, lecture 7. So this was an experiment that Dr. Azizi talked about, and it basically proved that ATP is necessary for a cross bridge initiation and termination. So as you can see from the first graph, when we first have ATP, but we don't add any calcium, it's going to be at a baseline level, because without calcium, the contraction would never be able to happen. But once we add calcium in the presence of ATP, we can see the tension rise. But then once we remove calcium, it'll drop all the way back down because it will undergo relaxation. So if you think about what happens from a biochemical standpoint, the calcium is facilitating the contraction, but then once we remove it, we're essentially putting it back into the sarcoplasmic reticulum. And so if you think about this from what would happen in a regular human body, like endogenously, this is what you would see. So in the presence of calcium, you would see high force, high tension, and then once you remove calcium from the system, the muscle goes back to baseline levels and it relaxes. So ATP is needed to initiate contraction and initiate and terminate cross-bridge cycling. But by itself, it won't cause tension because calcium is needed. And then in this experiment here on the bottom, it's kind of similar. So we added ATP in the presence of calcium. And like we discussed, the tension went up. But then something interesting happened. We removed ATP while there was still calcium. And as you can see, we still stayed at a relatively high tension. And that's because the cross bridges have already formed. So once cross bridges have formed, it doesn't matter that we have gotten rid of ATP because that bond between myosin and actin is very strong. But in the absence of calcium and when ATP is added, so in those two conditions, what we see is immediate muscle relaxation. So it goes from a state of constant tension to calcium not being there, but ATP is added, and it goes all the way back down. And then once we add calcium back in the presence of ATP, tension goes back up. So when added in the absence of calcium, myosin detaches from actin. Okay, this is another question. Upon death, the body undergoes a state of rigor mortis, which is also permanent muscle stiffness. What physiological mechanism accounts for this? Right, Jose, please. Okay, so in that case, ACP is not available to be added to the actin-myosin crossbridge, which prevents muscle relaxation, so the muscle will stay in a permanent state of contraction. Okay, so there's an interesting relationship between sarcomere size and tension. So we actually find that the optimal size of a sarcomere is intermediate. And so this graph shows us the maximum tension is at the middle point. So it's kind of like a regular graph. So it peaks right at the middle. So that's when we have optimal overlap between actin and myosin filaments. So what do we see on the short end? So on the short end, the reason why it's suboptimal is that the Z discs run into the ends of myosin filaments. And then we can also see from this figure, if we look at number 1, the actin filaments are very overlapped. So if they're very overlapped, that really hinders muscle contraction because they're not able to go towards and away from the M line, that middle line, properly. As we see in the middle part, there's sufficient overlap between actin and myosin versus the short sarcomere where actin is just overlapped with itself. And then in the long sarcomere, it's way too stretched out. So we essentially see little to no overlap between the actin and myosin filaments, which will again result in no contraction. Okay, so another thing is the force-velocity relationship. So at heavier weights, the muscle contracts at a slower rate, which this is quite intuitive if you think about it. So if I pick up a really heavy box, I'm not going to be able to pick it up as quickly as I'll be able to pick up these glasses, right? Because these are pretty light versus a very heavy box. My muscle will probably take some time to generate enough force to be able to pick it up. So the at maximum weight, which is in this case it's 100 grams, we see isometric contraction, which is basically when the muscle doesn't really stretch much because it's not able to handle the weight. Versus when the load is 0, we see no force being generated, and we're at Vmax, which is maximum velocity, because it takes very little to no effort to, carry something that weighs 0 grams, right? And then in between V0 and Vmax, we see a curvilinear relationship where force decreases constantly with velocity. So when we have very high forces, we're carrying very heavy items, essentially, we're gonna do that it's gonna take a bit more time versus lighter items. Which is, by the way, the right end, sorry, Jose. So the left part of the graph, that's where isometric contraction is, and then more like to the right is what we would call isotonic contraction, because the muscle does stretch. So in isotonic contraction, there is stretching of the muscle, and in isometric contraction, the muscle isn't able to stretch. Okay. So again, still talking about the force velocity relationship. Muscles that contract slowly have plenty of time for cross bridges to attach and detach. Muscles that contract quickly don't have enough time for cross bridge formation. And so it takes time to attach, to perform the power stroke, detach, and get ready to attach to a new site. That process takes plenty of time. So if we're just carrying something very, very quickly, chances are those cross bridges are going to form, but they're going to be in suboptimal configurations. And so they're not going to be able to form the power stroke in an optimal manner, essentially. So the faster the velocity of contraction, the more cross bridges with suboptimal configuration. So we're fighting the capacity to generate force at this point. So as you can see from the figure to the right, and this was these were the figures that he used as well. You can see that if you see this odd, the odd one out here is when you actually when you try to do it fast, it'll actually go in the other direction, right? So it opposes, when we say fighting capacity to generate force, that's what it means, because it's in the suboptimal configuration, versus the left side where the muscle, the sarcomere, has enough time to contract and perform the power stroke and then detach again. You can see that all of the myosin heads here are in optimal configuration. Okay, so real quick, muscle twitches occur when the nervous system sends a single action potential via one motor neuron. And so here we have 3 different periods where this pretty much occurs. So we have the latent period, which is when action potential has occurred, but the force has not yet developed. So we're essentially preparing for the contraction phase here. And the contraction period is when force develops within the muscle fiber. And that's where we see the maximum tension here on the graph. And relaxation phase is when the muscle goes back to resting conditions. You can see it just going down here all the way to 0. And the relaxation phase is associated with how fast calcium can exit the cytoplasm and get back into the sarcoplasmic reticulum is what determines the relaxation phase. Okay, so we have 2 types of tetanus. We have fused and unfused tetanus. So unfused tetanus is when force will fall, but as you can see from this graph, so do you see where the S's are with the arrows? So we're applying action potentials, but they're pretty spaced out. So as the force falls, the muscle fiber is hit with another action potential, but it's not a smooth activation. So what we see is this unfused tetanus where we have the muscle goes back up, attempts to relax, but right before it relaxes, we hit it with another action potential. So we have this just cycle of like going up, but not all the way down. So it creates activation, it increases activation, but it's not a smooth activation, versus the fused tetanus, which is a smooth activation. And it's where the muscle fiber is hit repeatedly, very quickly with action potentials in close proximity, which doesn't allow for any relaxation. So the tension you can see here on the graph, it continues and it stays at this high level. And that's because the S's, as you can see, are in very close proximity with one another. Moving on. Okay, motor unit recruitment. So a motor unit consists of one motor neuron in addition to all the fibers that it innervates. So small motor neurons or small motor units are always going to be recruited first, followed by intermediate, and then the large. And small motor units don't contribute much to force, whereas large units contribute the most. But the reason why we recruit the large motor units last and the small ones first is in order to conserve energy. The small and intermediate units are much more energetically favorable to use. versus the large units, which requires much more ATP, and it's just not an efficient way of carrying out contraction over a long period of time. So small motor units also have the slowest fibers, and then large units have the fastest fibers. So the tension generated by the fast motor units will rise and fall very quickly. So as you can see from the graph, and this is also the graph that Dr. Azizi was referring to, is as soon as tension goes up, it will come all the way back down almost instantaneously. Versus when you compare that to the slow line here, the light blue color, so you'll see that there's a very steady, slow reach to the peak tension, and then it goes down very, very slowly. And that also as a side note, that has to do with fast motor units having way more calcium pumps that can quickly remove calcium from cytoplasm after contraction has happened. It's able to relax very quickly because it does have many more calcium pumps. And this is a chart that compares the different types of muscle or motor units. So you can, I would really take the time to go through it and understand what each column is saying and the different, like the differences essentially between the three. So the slow oxidative one is the small one. The fast oxidative glycolytic or fast oxidative is the intermediate. The fast glycolytic is the large motor unit. So again, as we discussed, the myosin ATPase activity kind of intuitively will be high in most high in the fast glycolytic. It says that it's also high in the fast oxidative, but I believe that it is highest in the fast glycolytic, which contributes to the ability of fast glycolytic muscles or fibers to initiate contraction and go through the contraction cycle very quickly because they have a lot more ATPase activity. Okay, lecture 8. This is just a joke that I found. So my sincere apologies for interrupting. I ate actin when I say that, but I have had my calcium eye on you for a while now. So I love corny jokes. So I apologize if that has hurt you. Okay, so smooth muscle. Smooth muscles have the slowest contractile speed. They line the outer edges of hollow organs, so that will be your kidney, your liver, your esophagus. All these internal organs will be lined with smooth muscle. So they're very unique in that they can change their own shape and the shape of the organs that they're controlling. They're organized into antagonistic layers, which essentially means if we look at the diagram on the right, we have this kind of this longitudinal layer and the horizontal circular layer. So when a longitudinal layer contracts, it gets shorter. But what that means is that the circular layer gets larger and it gets wider. So I think the example that they used in the lecture was a water balloon. So they were saying that if you stretch a water balloon horizontally like that, it'll get longer versus when you stretch it longitudinally, it'll get wider from the sides. So they're antagonistic. Next slide. So smooth muscle anatomy, we have the dense bodies, which are analogous to the Z-discs and skeletal muscle. We have contractile proteins that run along the filament fibers, and then these the filament bundles will shorten, and that's what causes the smooth muscle to change their shape and to contract and to relax. Essentially, like they're unique in that they take up. They change their shape whenever they're contracting, right? Like they physically change their shape. And then we have vercosities, which are expansions of the axon that operate as replacement for the axon terminal. So, and then I'll talk about varicosities a little bit more when we describe single unit and multi-unit smooth muscle. Okay, so which of the following is an example of a body tissue that contains smooth muscle based on what we said? All right, let's see the answer. Looks like you guys are getting all these right, which is great. Stomach, yay! And that's because the stomach is an internal organ, so it will be lined with smooth muscle. And good job for not falling for the heart one, because the heart has its own cardiac muscle. Okay, so single unit smooth muscle. So the autonomic nervous system induces an excitatory signal. In the depolarization event, it causes these varicosities to release the neurotransmitter. So like we said, the varicosities, you can essentially think of them as the axon terminal of the smooth muscle, right? So they'll release the neurotransmitter versus the axon terminal, where in regular neurons, where you have vesicles releasing their neurotransmitter. Here we have the varicosities releasing the neurotransmitter into the extracellular space, and they kind of wash over the entire smooth muscle layer, and they spread that neurotransmitter without really discriminating at all, like which area to spread it towards. Like we're gonna see that's gonna be a bit more selective with the multi-unit smooth muscle, but with the single unit, we're gonna see that neurotransmitter washing over the entire layer with no difference in concentration. And then the reason that happens is because of gap junctions, right? So gap junctions connect all the cells within the layer, and so once the signal reaches one place, they're able to immediately let that signal go to the next unit of the muscle. So in contrast, multi-unit smooth muscles do not have gap junctions. So the autonomic neuron, which is this long yellow looking thing that you can see here. So the autonomic neuron extends and it encompasses all cells in the layer. So whereas before we had the kind of the varicosities releasing neurotransmitter and then the gap junctions connecting the different regions, here we just have this one neuron varicosity that kind of gets in between each individual part of the smooth muscle, and it releases the neurotransmitter that way. And this is unique in that it allows recruitment of one part of the muscle and not another. So that that yellow thing that you're looking at, it can just like kind of do a swerve where like, say like it doesn't want to reach one region, it can just choose not to do that essentially. It's kind of weird to think about, but I think the important takeaway from the last two slides is that multi-unit smooth muscle can discriminate between which regions to send the neurotransmitter to, in contrast to single unit, which is not able to do that. So membrane receptors and membrane channels both can trigger a contraction signal within the smooth muscle. And then we have what we talked about in lecture. It's analogous to the ryanodine channels in skeletal muscle, and it's IP3. So the IP3 receptors will take the role of ryanodine channels in this case in prompting the release of calcium channels out of the sarcoplasmic reticulum. And then we also have a unique feature here in these store-operated calcium channels that are actually able to sense a decrease in calcium stores in the sarcoplasmic reticulum. So once they sense that signal, they'll prompt an increase in calcium entry into the sarcoplasmic reticulum, which is important to prepare for the next cycle of contraction. So we have this unique mechanism in smooth muscle that does not exist in skeletal muscle. And this is just quickly going over the various steps in smooth muscle contraction. So as you go through them, you will see that they are quite similar to like regular muscle contraction. So like skeletal muscle. But the only difference is we see this calmodulin and then MLCK. So this is kind of the only difference, but essentially when calcium concentrations increase, calcium will enter the cell and is released from the SR, and then calcium binds to this molecule called calmodulin, and calmodulin will activate this kinase that's called myosin light chain kinase. And myosin light chain kinase is responsible for phosphorylating the myosin heads, and it'll increase myosin ATPase activity. And then active myosin cross bridges slide along actin, and you guys know the drill. Myosin and actin attached together form cross bridges, yada yada. I think I forgot to include the relaxation steps for this, but they are on your lecture. So I would definitely go over those and make sure you understand how they work. But it's essentially the reverse of these steps, right? So instead of MLCK phosphorylating light chains, you're gonna have MLCK or MLC phosphatase, which will take away the phosphate that the kinase put in, if that makes sense. So it'll dephosphorylate it, and it'll essentially decrease the activity and initiate relaxation. Don't know if I have anything else. Oh, which of the following molecules is needed for smooth muscle contraction? Okay, let's see the answer. And yeah, you guys got it. It's calmodulin. And that's it for my portion. Thank you guys for tuning in. Please don't forget to fill out the evaluation sheet. It really helps us know what we did wrong and understand how we can improve more for the next review session.
Yeah, good luck studying, everyone. We believe in you. I know this review session was an overload of information, but thankfully we'll be posting all this great work on the website.
Yeah, Andrew, good job. If you're that ready, I wish you could take it right now too. Good luck, everyone. Thank you for stopping by.
Thank you very much. Very helpful.
Of course, you're welcome. We hope to see you guys again soon, and you'll get through this. No worries. Take care.
Can you drop the website? How do we get to it again?
So you can use the QR code with your phone and camera. I'll also add it to the chat as well.
Okay, thank you.
Yeah, of course.
If anybody has any questions, yeah, feel free to stick around and ask it. You can unmute yourself or chat.
I have a question of when the slides will be posted, because I had a couple of questions, but it was just kind of hard to keep up. So
Sure.
I can probably ask one of you guys tomorrow or something.
Okay. However you feel comfortable. If you feel like you need us to go back on anything, we can try to do that.
Okay, for sure. But do you have an idea? Like, should we expect it to be posted tonight or tomorrow, probably?
We'll post the slides tonight, but we can't make any promises about the video since we still need to figure out the nitty-gritty part.
Yeah.
Okay. Yeah, the slides will be good. Thank you.
For Teresa.
Teresa, yeah.
I don't know anything about anatomy, but
Yeah. Crash course is good. It gives you good general knowledge. I feel Khan Academy, the MCAT section or the med school section, I think it's called Human Bio or something. But that I think is super in depth, super helpful. The crash course stuff is kind of like it highlights key terms and general ideas. It's good as like an introductory thing, kind of just to get your head thinking about these certain topics. But if you want more involved things like mechanisms, crash course unfortunately doesn't really cover too many mechanisms, and those are actually what most of these exams are based on.
I would say for myself, like having taken this course with Professor Azizi, I found that his midterms were very true to his lectures. So if you can go through all the lectures and understand pretty much every detail of what they go over, you're gonna be good to go. Like, I can attest to the fact that like he doesn't throw you like any curveballs or anything like that. So as long as you understand the lecture material, you should be okay.
Thank you guys.
Yeah,
No
of
problem.
course.
Thank you.
Thanks for coming.
Sweet.
Are there any more general questions, content related questions?
Yeah, I realized I made some mistakes. I look back and I'm like, oops, endocrine and exocrine glands. Yeah, so I saw you guys fix that for me. Thank you very much. Forgot what else I made, but I made a couple mistakes. I
I
tried.
do have one question about calmodulin.
Of course.
So could you go back to the contraction slide, please?
This slide?
So pretty much that with calmodulin, you need the presence of calcium for it to be activated to therefore then activate the MLCK.
Yeah, so think of it as a complex. So calcium binds to calmodulin, and then once that complex is formed, MLCK is able to come and do its job. Does that make sense?
And we don't need to know how it enters the cell, calmodulin itself.
No, you just need to pretty much know these steps and understand like their order of like what happens when, essentially.
Great. And then last question on calmodulin, is it a kind of all or none kind of response, or is it kind of graded?
I think it's just one of those things where once calcium binds to a receptor on calmodulin, I think that's the mechanism by which it acts. Like, so once that bond is formed, that's sufficient for MLCK to come in and and phosphorylate the light chains in myosin.
Great, thank you. Okay, awesome. That's all.
Thank you for your question. Does anybody else have any questions? Let's see. I can finally read the chat because it's not like there was a point where it said 33 people chatting. I'm like, I can't keep up with this.
I know.
I don't know how professors
There's
do
7
it.
of us now. So if you guys wanted to ask questions but were shy earlier, you can ask now.
Yeah.
Monica was so on point. Every time I like start typing, she did. I was like, wow, speedy fast.
Wait, sorry, your voice got.
Like whenever there was a question, you answered it super fast, which was nice, and I would be like in the middle, like, ooh, she did good.
Oh, and yeah, I was so worried about like missing something. I was like, oh my gosh, I hope I got to all the questions.
Yeah, thank you for staying and listening to us.
Yeah. Glad we didn't bore y'all. Seriously.
I know.
That was my biggest dilemma. I'm like, I'm either gonna mess up really bad or they're gonna be so bored I'm gonna see the numbers drop.
Okay, we're down to 4 participants.
Hello.
Can I ask you guys a question? I think I'm the last one here.
Yes.
Oh wait, you are.
You are. Woo, you made it to the end.
We got one-on-one attention.
I just have a question about the fiber type comparison. I believe this was in lecture 7 where he had the chart and he was comparing the small.
Wait, are we talking about? Hold on. Here?
Yes, right here.
Okay.
So I have a question about the calcium pumps in XR. Do you guys have that here? I don't think so.
No.
But in his lectures, he has in the chart he has the calcium pumps in the sarcoplasmic reticulum.
Yeah.
And so in the smaller fibers, it goes from fewer to many as it increases. I was wondering, could you guys explain why?
And the smaller fibers?
Yeah, so the smaller fibers have the fewest amount of the calcium pumps, and then the largest fibers have the highest numbers of calcium pumps.
I think it's because it's one of those things where you start off slow. So like you start off recruiting like just a few fibers, and then that causes an effect of like recruiting more and more. Is that what you're asking? Or sorry.
I think so. Now that you said it, it kind of makes sense. I think so.
Yeah.
But it's pretty much this chart. It's just like on his, he has like the CA2 pumps in SR, and then it goes in the order of fewer to many. But what you said makes sense though. So like the smaller fibers have fewer pumps, so they can't do. They're not as strong as the larger fibers, right? Is that what it is?
I think so. I think the important point with the calcium discussion was knowing that there's a lot more calcium pumps on the fast fibers. So because there's a lot more calcium pumps in general, they're able to go through the contraction relaxation phase much more quickly than the small and the intermediate fibers.
Okay, got it. And then my last question was about the endurance. It's on that same slide too. I just wanted to confirm, so with the larger fibers, they're easily fatigued. Is this because they're using glycolysis as opposed to the oxidative phosphorylation? So it's like less ACP being made?
Yeah, so the reason why oxidative phosphorylation is so much more cost effective is because you can use one glucose molecule and make a whole bunch of ACP. Perhaps I think now that I'm thinking about it, I think I should have explained that a bit more. I was just afraid of like being short on time, but yeah, that you're right. So oxidative phosphorylation is going to be way more effective than glycolysis because you can make way more ATP for not that much energy.
ATP, got it. Okay, thank you guys.
No problem.
Studying.
Good luck. Yeah.
Good luck.
I don't know why I just said YouTube.
We're studying too.
Oh, you are?
Yeah.
Okay, good. Good luck, guys.
I guess we're students too, right?
Yeah, you are.
Yeah, shoot. Almost felt like a teacher there for a second. Okay. Thank you.
Good night.
Hello.